And my name's Doug Record. I'm the principal of Bournda Environmental Education Centre, and I'd like to welcome anyone who's joining this video conference today or watching it later. I'd like to thank you for coming along, and I'd just like to acknowledge the traditional custodians, the elders past, present and emerging, and to extend those acknowledgements to any Aboriginal persons who watch this video conference. And I guess in dealing with University of Otago and our guest, Associate Professor Ceridwen, Fraser, we've got a lot to learn from New Zealand in terms of incorporating Indigenous, making that fundamentally part of the way people do things. It's always good to get those emails from the conferencing team at Otago with what's the tagline on those emails? It's a greeting in Maori. Might be ngā mihi or kia ora. One of those. So it's always a nice thing to see that acknowledgement of culture. So our guest today, Associate Professor Ceridwen Fraser, Rutherford Discovery Fellow at the Department of Marine Science at the University of Otago, also known as Crid, and we'll refer to you to Crid as Crid, if that's all right today, Crid. Of course. And we're joined by a few guests from Bournda Environmental Centre, teacher Julie Don and teacher Luke Brown. And we got eyebrows from Luke. He's the one with the Hoff behind him. So, look, it's a pretty relaxed sort of a chat, and it literally is, if I point my camera at the fireside, it literally is a fireside chat with Crid to hear about what work she's been up to, and to be the first in one of 3 online meetings today to celebrate World Environment Day. The theme this year in 2020 is For Nature, and there's a very strong biodiversity message in that. And I guess today, kicking us off, Crid, you're leading the way in telling us a little bit about how the research that you are conducting is making a contribution for nature. So I'll hand over to you, and we'll go through your presentation, and then any questions that come up, we can have a bit of a chat about after that.
Cool, thanks, Doug. Very nice introduction. And yes, stick with calling me Crid, but in New Zealand we say crud. So the stuff on the bottom of your shoe. But yeah, so Doug's direction for what to talk about today was fairly open, and so I thought I'd go through some highlights from some of my research. I think they're the highlights. It's mostly about kelp because, as you can see in this photo, I'm fairly obsessed with kelp. A marine biologist in the UK said yesterday that he thinks I need an intervention. I'm getting too obsessed with kelp. I made a very nice wakame salad on the weekend with some invasive undaria. So keep an eye out for that along your coastline, because you can eat it and feel good about saving native species at the same time. But one of the things that I'm really interested in my research is long distance dispersal. How do plants and animals travel long distances? Do they? Where do they? And what does that mean for patterns of biodiversity? And Charles Darwin was thinking about this back when he wrote The Origin of Species. And he was saying, you know, we see all these similar species in really distant places around the Southern Hemisphere, like in the Kerguelen Islands and in South America and in New Zealand. We see very, very similar plants and animals. So how can that be? And his idea was that maybe icebergs were carrying plants and animals across the sea, and that could explain how there are similarities between the continents. Because of course in those days they didn't know about plate tectonics. So where countries or where continents were was where they had always been. And that might seem crazy that icebergs could travel across oceans and carry plants and animals with them. But not long after I started my PhD back in 2006, and I did it here in Dunedin, even though I'm Canberra born and bred, I've fallen in love with New Zealand, and so I did my PhD here more than a decade ago. But just when I started, there was this exciting moment because a huge iceberg that had broken off in the Weddell Sea in Antarctica a few years before had drifted right around in the path of the Antarctic circumpolar current and had come up past New Zealand. So we could see it from Dunedin, standing on the shore here. You could see an iceberg drifting past. It was pretty cool. It was a huge iceberg. And then the Australian Channel 9 flew a helicopter. You can just see it in that picture from the newspaper, little helicopter landing there. So the Australian Channel 9's helicopter came over and they planted an Australian flag on the iceberg. And you can imagine how the Kiwis reacted to that. There was outrage. So in retribution, the Kiwis flew their most famous sheep over to the iceberg. He was called Shrek, and he had little crampons made so he wouldn't slip on the iceberg. And they flew this sheep over to the iceberg and had a ceremonial shearing of Shrek, and thus reclaimed the iceberg for New Zealand. But this demonstrates that icebergs do travel quite long distances at sea, and that things like ocean currents, in this case the Antarctic circumpolar current, can carry floating objects long distances. More recently, there's been research that's shown that a lot of the vertebrates that live in Madagascar, you know, Madagascar is famous for having really unusual animals like lemurs, but a lot of the animals in Madagascar have not, the estimate of how different their DNA is to similar animals in Africa shows that they probably haven't been separated from African relatives for very long, certainly not as long as Madagascar has been separated from Africa. So researchers now think that a lot of the vertebrates in Madagascar rafted over, perhaps floating on bits of driftwood, from nearby Africa to reach Madagascar. So I've spent a lot of my career working on and off with this really charismatic organism, not the seal, but the much more exciting kelp that it's swimming through, which is Davillia antarctica, or really Davillia because we've split Antarctica into a whole bunch of different species now. But this is a buoyant, floating kelp that grows around much of the southern hemisphere. It's a really important species for ecosystems. You can see here it's washed up on the beaches in the sub-Antarctic. It grows all around the intertidal in the sub-Antarctic, around New Zealand, around South America. There is a sister species in Australia, but it's a non-buoyant one. It's slightly different. There's actually 2 species in Australia. So as I said, it floats really well. You could almost lie on it like a lilo, and it's very large and very robust. So it's kind of an ideal raft making material. And in fact, Maori did make boats out of this kelp at one point. Someone estimated that there's probably about 70 million rafts of this kelp drifting around the Southern Ocean at any time, and about 20 million of those still have the holdfast attached. That's that chunky bit at the bottom that sticks onto the rock. And that's important because the holdfast is home to heaps of different animals. It's home to snails and worms, bivalves, chitons, crustaceans, echinoderms. All sorts of stuff is living inside that holdfast, and so could drift at sea with the kelp across the ocean if the kelp can really travel a long way. And so that was kind of the focus of my PhD. And then towards the end of the PhD, I found some really nice evidence that kelp really was carrying whole communities across oceans. I was walking along St. Clair Beach in Dunedin with my husband, and we came across some really exciting kelp. My husband doesn't go for romantic walks on beaches with me anymore because he always ends up carrying slimy bits of kelp back in his pockets. But the kelp was exciting because it was covered in huge goose barnacles, and goose barnacles are these kind of stalked, tear-shaped barnacles, and they only settle, some species only settle on things that are drifting out at sea. So if you find large goose barnacles on something washed up on the beach, it means that object has been drifting at sea for a very long time because the goose barnacle must have grown on the object drifting at sea. So we knew that this kelp had travelled at sea for a long time. And where had it come from? We could estimate kind of how long minimum it had been at sea by the size of the goose barnacles that were growing on it. So based on the length of the capitulum, which is the shell part of the barnacle. Some of the red crosses on this graph show the size of the largest goose barnacles on each of the kelp rafts on the beach that day, and the blue squares are known growth rates of barnacles of different ages. So we can see that the largest goose barnacles on one of the rafts had been at sea for at least 2 months, probably more. And then doing genetic tests, we were able to show that that kelp raft and the animals in it had come from the sub-Antarctic islands, at least 400 kilometers to 600 kilometers away. So we were able to use DNA to kind of indicate which population it came from. Now, at the time, the DNA methods I was using weren't super high resolution. They've become a lot more powerful now. So we could kind of say it was either the Snares or the Auckland Islands, but we couldn't say with any great certainty which one it had come from. But certainly it wasn't local. It had been at sea and had travelled hundreds of kilometres, and it still had holdfasts packed full of living animals. So that was kind of cool. It was the first evidence that communities, whole communities, ecosystems could really travel at sea, floating like this with kelp rafts. So we knew then that rafting could happen, that it was really effective, but we didn't really get a sense of just how far could the kelp travel, how big could these rafting events be. And then a couple of years ago, in 2017, I got an email from a colleague, and he said, "Hey, have you ever seen your bull kelp in Antarctica?" And I was super excited. He took that photo on the left there. That's a big chunk of bull kelp lying in the middle of a beach in Antarctica. But everything we knew about Antarctica and the ocean around Antarctica suggested that kelp should never have been able to reach that beach, that it was an impossible voyage. Because Antarctica is really isolated, it's surrounded by ocean, it's surrounded by strong currents. And although things like seals and penguins and flying birds and whales, you know, strong swimmers and strong flyers can get in and out of the Antarctic, there's no evidence of any other smaller plants and animals kind of getting into and out of the Antarctic on their own. So we thought Antarctica was isolated and that nothing would be able to cross the Southern Ocean to get there. It's not just that there's an ocean in the way, it's that the currents of the ocean are the strongest current. It's the strongest current in the world, the Antarctic Circumpolar Current, and it goes eastward around Antarctica, pretty much unbroken. And then there are also these polar fronts that you can see here, these dotted lines that circle Antarctica. And those are thermal boundaries where cold water meets warmer water, which creates a subduction zone where the water bodies meet each other and go downwards. So anything floating at the surface was thought to not be able to cross these subduction zones, these barriers, the thermal fronts. So we didn't think anything could go from the north to the south. But here in yellow, I've circled the distribution where we find De Villiers Antarctica, this floating bull kelp. It's all around the sub-Antarctic islands, and it's all along the west coast of South America, and it's all around the coast of New Zealand, but it doesn't grow south, much south of the Antarctic polar front, and we didn't think it would be able to cross south to reach Antarctica. But we found the kelp washed up on a beach at the tip of the peninsula here. And like Doug said, just jump in with questions if you guys want to. Don't feel like I'm just rabbiting on. Well, I am, but you can interrupt. So like I said, DNA analyses have come a long way since I did my PhD, and we now have much more powerful tools. And we were able to resolve exactly where the bits of kelp that were found in Antarctica had come from using DNA analyses that could show differences between populations. So even within South America, for example, the stuff from Patagonia, like this central bit here, was distinguishable from the stuff from Cape Horn, the bull kelp DNA. And even within the Falkland Islands, one site in the Falkland Islands is distinguishable from the other site. So using these really powerful DNA methods, we can really see differences between populations, even really close together populations. And that meant we were able to say that one of the pieces of bull kelp that was in Antarctica had come from the Kerguelen Islands here. and one piece had come from South Georgia here. So they'd travelled a very long way. And I worked with some oceanographers to try to work out how this was possible, because what we knew of ocean currents suggested that it wasn't possible. And the oceanographers, originally when they started to try to model how could we get the kelp from where we knew it had come from to where we found it, they couldn't do it. The existing models just didn't work. But we knew it was possible because we'd found the kelp. So they kept at it, and finally they realized there was something missing in their models, and that was the effect of wind creating massive surface waves, which kind of push the kelp over boundaries. So it's not just the surface currents, but the surface waves, where a wave will pick a particle up and push it forward, and then the next wave comes along and picks that particle up and pushes it forward, which is separate to the surface currents that they were modeling. And when you incorporate that surfing effect, then you can start to get kelp reaching Antarctica in the models. So in this movie, the orange-tailed particles, this is 140 particles all released from South Georgia at the same time. You can see they all take completely different trajectories. The ones with orange tails are eventually going to reach Antarctica, and the ones with blue tails are not going to reach Antarctica. So there's a huge spread in the paths that these particles take. They go all over the place. And it just goes to show, when you throw a message in a bottle out at sea, you can't really predict with any great certainty where that bottle's going to end up. It could take any of a whole bunch of trajectories. But the cool thing about this modelling was that it showed that we could get particles coming from South Georgia or Kerguelen Islands and hitting the Antarctic Peninsula, where we found those kelp. So this kind of overturned what we thought about Antarctic isolation. So the shortest simulated trajectory from Kerguelen Islands was 20,000 kilometres, which is a massive distance for kelp to have travelled. The average from South Georgia was 25,000 kilometres, because even though South Georgia is quite close to where they were found, the currents go this way. And so all of the kelp particles set off in this direction. Some of them took the long way round in the simulations. Some of them got about this far and then were caught up in the Antarctic gyres and came back along the coast this way. But in any case, none of them were taking less than 9,000 kilometres to get from South Georgia to where they were found. So that's a phenomenal rafting distance. And it's the longest rafting voyages ever recorded. And so even though it was only a tiny proportion of the simulated particles that reached Antarctica in all of these models, tiny, tiny proportion, when you think that there's estimated to be 70 million kelp rafts drifting in the sea at any time, that would mean that if every kelp raft is hitting Antarctica within 2 years, which most of the models showed was feasible, then we're probably getting somewhere between a handful and several thousand kelp rafts hitting the Antarctic coast every month. So that's actually like a huge influx of plants and animals from the north that don't currently occur in Antarctica. So we now know that floating objects, including kelp rafts, can and probably often do reach Antarctica from the north. So Antarctica is not isolated, and that also has implications for Antarctic ecosystems beyond the intertidal marine kelp zone, because of course those same processes can transport anything that's floating right at the surface, like plastics and plant seeds, into the Antarctic. So at the moment, the Antarctic's too cold for these species to establish, but it's one of the fastest warming parts of the planet. And there will be parts of Antarctica that could be hospitable to these organisms by the end of the year, but not by the end of this year, by the end of the century, sorry. We're expecting that parts of the Antarctic Peninsula will be similar to sub-Antarctic environments. And so then you might start to see these big sub-Antarctic kelps settling and growing in Antarctica with the animals that they carry too. So is it a problem if kelp carries passengers to new places? Well, it's a problem for Antarctica because Antarctica is isolated and this is going to change the ecosystems. It's probably quite good for the kelp because it's going to be able to change its distributions. But kelp also carries pathogens like parasites and diseases with it. So there's scope for this rafting events to carry pathogens quite long distances. And I thought I threw in this little study because it does link back to the south coast of New South Wales. When I worked in Australia at the Australian National University, I had an honours student one year, Callum, who did some really interesting work looking at protistan pathogens, so a disease of the kelp, which creates warts. You can kind of see these warts all over the kelp here. Those are not normal. They're caused by protists, which are kind of single-celled organisms, a bit like bacteria. And Callum did some ecological surveys along the Australian and the Chilean coasts, and he also did some genetic analyses of these pathogens. So he collected some samples at Tathra, and one interesting thing was he found that we were kind of expecting that at the northern limit of the kelp's range, which is pretty much Tathra or thereabouts, that the disease would be greatest in those populations, because those are the populations that are kind of most under stress. They're the ones that are struggling because it's kind of too warm for them, and that's why the kelp populations are starting to head south along the coast. But in fact, he found the opposite. He found most of the infections were down in the south, in the central part of the kelp's range. But when he did some genetic analyses, he found that there's a very strong genetic break in the parasite or the pathogen across Wilson's prom, which is interesting because that's a biogeographic break for a whole heap of Australian marine species. We see genetic differences on the west side to the east side of Wilson's prom. We think it's Wilson's prom, but of course this is kind of mostly beach along here. And most of the species we've looked at are rocky shore animals and plants that don't occur on the beaches. So we're not quite sure exactly where the break is, but we think it's Wilson's prom because that was the last point of connection of the mainland to Tasmania as the waters were rising after the last ice age. So we think that at the last ice age, there's this kind of barrier between Tasmania and the mainland for marine life on this side and the marine life on that side. And then when the waters started to come up and that barrier disappeared, things were able to move from east to west or west to east again. But it didn't really make much difference because there were already this genetic differences. So Gurdeep, you're asking, why is it only kelp rafting? Instead of what other sorts of things? So I can't hear you, you're on mute.
Yes, can you hear me now?
Yeah.
Yeah, I was just wondering why it's only kelp. What about other things? Did they have found other thing, or it's only kelp they have found in Antarctica that floating that far away? Is it any other?
Yeah, so it's interesting. I mean, there are lots of other things that float, and I mentioned plastics and seeds, and we have been finding a lot of plastic in Antarctica, we being scientists, that there's a lot of findings of plastics in Antarctica, and everybody thought that those probably must have come from ships visiting Antarctica, but there's so much plastic in Antarctica that it's hard to explain just from the few ships that go down there. And so now we think that this rafting or this drifting surfing effect probably explains why there's so much plastic reaching Antarctica. And then of course things like driftwood can travel too. I don't know of driftwood washing up in Antarctica. I'm not sure if anyone's recorded that, but certainly when I've visited the Falkland Islands, which are kind of east of South America,
yes,
you see driftwood there of plants that don't occur in the Falkland Islands that are probably coming across from South America. Oh. So yeah, driftwood and kelp and plastics and really anything that floats can raft. Pumice is another really good one.
Which one, sorry?
Pumice, like you know, the rock that you use to grind dead skin off your feet. Yeah. That's created by volcanoes, but it's very bubbly. So even though it's a rock, it floats really well. And a lot of animals can sort of live in it because it's got little holes all through it. And it can raft long distances across oceans too, carrying algae and animals with it.
Oh, yes, that's interesting. Yeah. Thank you so much. Thank you.
No worries. So yeah, Callum was, he found this genetic break in the pathogen, and it's interesting because it kind of lines up with the two different species that we have in Antarctica. So there's a different genetic signature for the pathogen on one species versus on another species. And in some cases, like the pathogen that hits the Western species is more similar to the pathogen from Chile than it is to the pathogen from kelp in other parts of Australia. So that was really cool. Okay, so we know that kelp can travel a really long way, and is this ever a good thing? Well, it's really useful for recolonization after a disturbance. And so this is a nice example from New Zealand. You might remember a few years ago in 2016, there was a massive earthquake, 7.8 magnitude earthquake centered around Kaikoura area in North Canterbury, which is the sort of northern part of the South Island. And it lifted the seabed a phenomenal 6 meters in some areas. So this is a subtidal seaweed that normally grows only underwater. But this entire rock platform came up 6 meters in the blink of an eye during the earthquake. So all of these rocks were underwater just a few, actually this photo was taken a couple of years after the earthquake, but within seconds, this entire rock platform had popped out of the ocean with all the subtidal seaweeds. There were dead fish lying on top of the rocks, lobster pots sitting out here. It was bizarre. We got in there 5 days after the quake, and it was like walking through an underwater world, but without the water. It was mind-blowing. This is this area of rock platform. You can see the fault there, where all of the brown stuff is seaweed dying in the sun just days after the quake, because every bit of brown would have been underwater 5 days before this photo was taken. But it split along this fault and it popped up. And you can see there, this is the road we came along. The road had come up several meters. The railway line was up in the air. It was really crazy. So it wasn't 6 meters all along the coast. These photos were taken at Waipapa Bay, where the uplift was greatest. But at other sites, even tens of kilometers away, you still had uplift of 1 to 3 meters along most of this North Canterbury coast. And so that completely wiped out the bull kelp intertidal communities, because they obviously can't move. And they were left high and dry and stranded. So this is 5 days after the earthquake. Again, subtidal seaweeds that would normally be underwater. And this is 5 months after the earthquake. You can see it's all just died and fallen off, and there's nothing left at the intertidal zone of those big intertidal habitat forming species that are so important for ecosystems. So this coastline has changed drastically now. The species that kind of make it what it is are gone. But we know that kelp can raft. Oh, these are just one more photo. 5 days after the earthquake, all the big bull kelps stranded up on the rocks. 5 months after the earthquake, no sign of them. They're all gone. So kelp rafting will enable that coast to be recolonized. So if the kelp couldn't travel, then it wouldn't be able to recolonize this coast. Once it's gone, it's gone. But I had an honours student a couple of years ago who walked along the whole stretch of the North Canterbury coast, picking up bits of bull kelp washed up on beaches. And she did these high resolution genetic analyses to work out where they came from. And some of them were coming from thousands of kilometres away. So there's this constant supply of rafts and animals, of kelp and animals, coming from hundreds to thousands of kilometres away that are hitting this coast, which gives it a really good chance of coming back in a few years. That the kelp can't stick back on, but the kelp that's hitting the coast is still reproductively viable, it's still able to produce eggs and sperm. And so it can have babies that will colonize that stretch of coast and will kind of seed the new ecosystems. And so that's really, really important mechanism that kind of lets the kelp find new places and establish and grow and ecosystems recover. So finally, I just wanted to talk about a way that we use DNA these days. I'm finding a really exciting new approach. So I've talked about using DNA to kind of work out where things came from, but we can also use DNA just to characterize communities without much knowledge about those communities beforehand. So this is environmental DNA. Have you guys heard of environmental DNA? et al., or eDNA. It's a great approach. It basically takes the idea that in an ecosystem, in any given ecosystem, every living thing in that ecosystem is producing and shedding DNA. So in this ecosystem, we've probably got some fish swimming through the water, some little crustaceans and worms hanging around in the sand and on the rocks. There's mammals, humans and sea lions. There are birds flying past. There are trees along the coastline, shrubs. There's seaweed. There's all sorts of plants and animals in this ecosystem. And every time you do something, like you move, there's bits of your skin that are falling off into the ecosystem. When you eat something, you're dropping crumbs of what you're eating into the ecosystem. When fish swim, they're doing poos, they're shedding DNA into the ecosystem. So there's DNA of everything there in that ecosystem. So all we have to do is take a sample of the water or the sand and analyze the DNA in it to work out what lives in that ecosystem. And that's different to the way we normally characterize ecosystems, which is kind of laboriously looking down a microscope and trying to figure out what things are. I did my honors in this back in 2005, and we dug up some mud from Botany Bay, and I tried to analyze the worms that we found in that mud to see whether there was an impact from the dredging that took place to build the new runway for the airport. And in that project, we collected nearly 4,800 polychaete worms, marine worms, which were from 32 different taxonomic families across more than 100 different species. So I spent most of that year peering down a microscope, trying to figure out how many hairs were along the side of the worm and whether it had this tentacle or that tentacle, to figure out what species it was so that I could characterize that ecosystem. And that was just the worms, not even considering any of the crustaceans or other things that might have been in that mud sample. So that's a very time-consuming process to work out what's in an ecosystem. And it requires a heap of specialized knowledge. Like, you need to know everything there is to know about worms before you can figure out which species is which. So eDNA instead gives us the opportunity to just take a sample of sand or soil or mud, and then look at what DNA is in it, and that will tell us a whole lot about the community. So here's an example of how I used eDNA in a recent study. I was interested in whether volcanoes were supporting life in Antarctica through past ice ages. There's a whole heap of volcanoes that have been active during the recent ice ages, and they form clusters in Antarctica. There's also some really hot granite rocks. And the idea was that these volcanoes might have created places in Antarctica that plants and animals could survive even when the rest of the continent was completely covered in ice. And whether those places were on top of a volcano or so you can see here, this is a volcano I visited a couple of years ago in Antarctica. Even though there's ice all around, the soil is like 45 degrees, and it can create some sort of ice-free areas that plants and animals could survive. But you also get caves hollowed out by the volcanic steam under the ice that could be fantastic places for plants and animals to hang out during an ice age. But if that's the case, if plants and animals survived in subglacial caves under the ice during ice ages, then we should still find plants and animals in those caves today. And so I got some soil samples from caves around Mount Erebus, a volcano in Antarctica, and used an eDNA approach to try to find out whether there's evidence of plants and animals in those environments. And we found in Harry's Dream, in one of the caves, we found evidence of algae. In Warren Cave, we found evidence of springtails, which are little Antarctic insect type things, mosses and algae, and various worms, oligochaetes and nematodes. So that was really exciting. We found evidence of plants and animals down in the caves that we would normally find in other parts of Antarctica. And in another cave on the other side of Mount Erebus, we found evidence of some unknown arthropod. And when I say unknown, it means we couldn't match the DNA from that arthropod to anything in existing database of sequences. It doesn't mean it's not a new, it doesn't mean it's necessarily a new species. It just means we couldn't match the DNA code directly to anything we've ever sequenced before. And the problem with that is it led to all sorts of media excitement, because when the media were quizzing me about this, I was saying, oh yeah, we did find some sequences that we couldn't really identify properly. And all of a sudden there were headlines like, aliens might be living under the ice of Antarctica, which is not what we said. But, you know, in a way, I can't see why there wouldn't be species that we haven't discovered yet living in those cave systems. It seems entirely plausible. And there's like more than 100 volcanoes in Antarctica that are lurking beneath the ice, must be creating very extensive cave networks under the glaciers. Yeah, as Doug says, this is like, media just love this sort of thing. And I don't really mind so long as the facts are right under the crazy headline. It got people paying attention anyway. But the interesting thing about eDNA is, so yes, we found evidence of Antarctic plants and animals in the caves, which is exciting because it probably means there's living and plants and animals in those caves. But that's yet to be confirmed, of course, because all we have is DNA. But we also found DNA from North American trees and soybeans and kiwi berries and all sorts of other foods. So this was, at first I was really frustrated thinking, oh no, you know, it kind of screws up our results, because how can we trust our Antarctic and plant and animal DNA when we're also getting all these crazy foods and woods and things showing up? But I was very confident that the DNA that we were sequencing was from the caves in Antarctica and not from the lab, because we had a whole bunch of controls in place that would have shown if it was lab contamination. So these were DNA results from the caves in Antarctica. And that got me wondering about why you'd find North American trees in Antarctica. Nobody really takes wood down to Antarctica these days. Most of our equipment is made of fiberglass and titanium and that kind of stuff, and plastics. So, but back in the day, people made skis out of ash, and ash was the tree that we were finding in the DNA in the caves. And we also picked up black locust, so another North American tree that was a hardwood that was used for things like ship nails back in the day. So I wondered if the foods and woods that we were finding in the caves might link right back to the first explorers, Scott and Shackleton, who went up Mount Erebus more than 100 years ago. And I spent a long time kind of trawling through their diaries and looking through their supply lists, and everything, every food stuff that we found evidence of in the cave can be linked back to the supply lists of either Scott's group or Shackleton's group. And with Scott's group, they mapped their journey up Mount Erebus. And if we orientate that, the red line shows the route they took to go up Mount Erebus. And you can see that they basically walked right past Warren Cave, where we found all this stuff. And I think it was Shackleton's group lost a whole bunch of materials, like a whole sled of supplies, while they were up the top there. So I never confirmed this, and I don't think we ever can confirm that it was a link to the first explorers, but it could be, because if anywhere can preserve DNA for more than 100 years, it's Antarctica. It's just a giant freezer. So I think that's quite exciting, that there could be genetic evidence of the first explorers still in Antarctica that we could use to answer some really interesting questions about where they went. And that's really it for my research. And I just wanted to finish because Doug, I think, was going to mention a little book that I'm writing. And this was my lockdown project. So when we went into lockdown, I needed something I could think about that wasn't too stressful, that was kind of something fun to work on, that made me feel like I was doing work, but wasn't too hard on my head. And so I decided to write a book about the interesting things that wash up on beaches. It's meant to be a guide to beachcombing in the Southern Hemisphere. And it's kind of looking at weird stuff that washes, weird or interesting things that wash up on beaches, and what that tells us about life and death in the sea and ocean currents. And it was my lockdown project, and I'm back at work today. So I've actually already sent it off to the publisher, but it's going to go under peer review now. So I just wanted to put out a call. If you have exciting photos of cool things you found on beaches that you'd be happy for me to include in the book, or stories about weird stuff you've found on beaches. Like somebody I came across had found part of a rocket ship, and other people have found ambergris and all sorts of cool things on beaches. So yeah, feel free to flick me an email. And yeah, let's have a chat. Sorry, it was mostly me talking so far.
Well, that's fine, Crid, because it's all been wonderfully interesting and a really good presentation. And I just sort of we've got Gurdeep. You're a high school biology teacher. I'm not sure. I know you picked up the link on the science notice board. Are you teaching in a high school?
I'm doing bio and investigating science and chemistry. So I am like doing everything little bit. And for investigating science, I like this concept of peer review and how the investigation has to be reviewed. And for bio students, it's really a dispersal of organisms. It's a really interesting. So thank you very much, Crid. It was really great presentation.
Thanks, Goody. And yeah, your point's really good. So a lot of what we as scientists produce, almost everything we publish has been peer reviewed, which means it's sent out to other scientists for them to check. And that can be quite confronting because sometimes they come back and say your experiment was bad, fix it. And sometimes you can and sometimes you can't. But it means that when you see scientific results published in peer-reviewed journals, then you can be confident that that they have been carefully checked by other scientists who weren't involved in the study. It's not entirely foolproof, but it means that you can kind of trust those results much more than if a scientist was just saying, look, we did this stuff and we found this cool stuff. It's all been through a process to try to make sure it's as accurate as possible or as robust as possible.
Yeah, and we tell students about validity and reliability of the resources, which adds to your point. Yes, yes. It's valid and reliable resource. So we talk about secondary data, which is reliable. But the photographs were beautiful of earthquake. Oh, that's amazing. Thank you so much.
It was pretty mind-blowing. Yeah.
Yes. Yes, yes.
And Jackie, what was Jackie's question, Doug?
Look, I'll just introduce you. You've met Jackie, haven't you, Crid?
Possibly. I can't see her face, so I'm not sure.
All right. So Jackie Randalls, for the other people in the meeting, is the CEO of Inspiring Australia New South Wales, which is a organisation based at Sydney Uni and working on science community engagement. And Jackie's been pivotal in supporting the Sapphire Coast Regional Science Hub Sustainability Education Network. And I'm really glad you're here, Jackie. Can you...
I'm so sorry. I had my computer down because I'm on a screen and I only just managed to jump on. But I loved that beachcombing idea, Crid. What a fantastic project.
Thanks. Yeah, it was really fun to just go through. There's lots of bits about different animals that you might find washed up on the beach, sea urchin tests and mollusk shells and that kind of stuff. But it did make me really appreciate the huge diversity of stuff that washes up on beaches. And I think everybody of all ages likes wandering around beaches and poking at things.
Oh, don't we ever? Although we've had the shipping container disaster in New South Wales and we've found plastic containers and face masks all over Sydney beaches.
Oh,
it's actually really depressing.
Yeah, somebody mentioned that to me.
It's been horrific, particularly when we've got this global pandemic, and these are all important safety equipment.
Yeah, well, we're having that problem all around the world where people are using masks and gloves and then just taking them off and throwing them
all over the street. It's disgusting.
Yeah. Now, Crid, Gurdeep asked a question before privately to me, which was, could we have a copy of your PowerPoint presentation? And I boldly said yes. Is that okay if we get a copy of the actual PowerPoint?
Yeah, that's fine.
Thank you very much.
Thank you.
And
you
look,
might have to remind me.
I think, as we said in the introduction, the purpose of these webinars, and there's what happens here, but the fact that it's being recorded by University of Otago and there's a resource there means that there's a longevity to the use. And I just Crid and develop that relationship, because I think it's a very important one for schools. And I think it was through a CSIRO program, the Science Professionals in Schools, or CIS program it was called, something along those lines. And we actually put our name down as a school, and CSIRO, if that was the organisation, linked us up with Crid. And I have to say, Crid, we've always you're a really good science communicator, and it just has been a really nice link for us to have. And if anyone is in a school, I think being part of those STEM professional programs is a really positive idea. And it allows you to just, get access to, academic researchers and build that relationship, and it becomes more a conversation than just consuming material that you're finding on the internet. So thank you, Crid, for being part of that program.
Thanks, Doug. It's been good both ways, I think.
Yeah.
Crid,
Great
it's
to meet everybody.
Crid, it's Luke here. I was just going to say we had some really interesting rafting kind of events happen a few years ago when we had the volcano that occurred off New Zealand, and then a lot of the pumice travelled through the Pacific and then ended up on our east coast.
Yeah, that's right.
So we were able to collect some of that and actually in the aquarium keep alive tropical anemones and polychaete worms and things like that were definitely not from our area. But all these sort of tropical species that they'd sort of picked up on the way. So that was really interesting.
Yeah, it's really cool. So I had an honours student working on that at the time too, looking at whether the size of the pumice clast affects the amount of organisms that it can carry. But yeah, there was like millions and millions of tons of that pumice drifting around in the ocean after that one eruption, and covered in plants and animals, or algae and animals. So really cool.
It was just alive when it got to our part of the coast.
There's some suggestion that that's maybe how the Great Barrier Reef got started, was with arrival from tropical species that came on pumice.
Yeah, it was, we were able to keep some of those things alive for months in the tanks too, just floating around.
Cool. Karen Woods from the Marine Centre there sent me some photos of the pumice washed up on beaches for that beachcombing book. But yeah, if anyone has any other nice ones of the animals on the pumice, I'd be keen to get one or two.
Yeah, I think there was some.
I have a kind of out of focus and.
Yeah, there was some shots taken, and just trying to identify what it actually was that it was growing on there.
Thank you, Doug. Thanks.
Look, a final thank you, Crid. Crid is also a mum and a wife and a working scientist, and that must be hugely demanding. And we really just appreciate your time. The photos were fantastic. I love the quality of the graphics, and I'll get everyone to put their hands together, and thank you very much for your contribution to World Environment Day.
Thank you.
Thanks, Doug, and thanks everybody for coming along.
All right.
I'd love it if you take
Crid.
this back to students in schools so that it gets out to a bigger group too.
Yeah, and thank you to the University of Otago e-conferencing team.